Hi, good morning. So I've posted the slides for today in the on the Learning Hub, so you could pull that up to follow along. Just in case the slides get stuck again, you could follow along that way. Okay. Please let me know if you see it. And I've also opened the chat, so you could answer questions via the chat. For those who are still not on Facebook, I will pull up the poll everywhere. I'll do that now. Okay, so let me know if you're seeing the chat. Just indicate at least one person indicate. Let me know if you're there. I am not seeing you. Well, let's see here. Okay, so please. Okay. Okay, it's now 10:32, so we should get started. One second here. I think I have, let's see, Tony's here. Okay. I'm just acknowledging who's here. And then we'll get started. Oh, just a couple things to address. So the test is still on Monday, regular time. And tomorrow you have your presentations. The first group will have their presentations tomorrow at 2 lab time. So someone asked me what should they bring to the presentation. Just bring your presentation. The actual paper, the research paper, is due on the day of the final. So you have time to still write that up. So bring that on the day of the final. Yes, Rayford, yes, you are in the chat. Okay. All right, so I asked you guys to look at some figures. For homework, study a couple of figures, well, 3 figures. And I will start there in the lecture, and then I have some questions related to the figures, and we'll see how we could answer those questions using this format, okay? All right, so let me get to that slide. Yes, Kimberly and Crystal and Clementine, I see you in the poll. But if you could actually get to the Facebook chat, it's just a lot more convenient. But if you don't have a Facebook page, we'll deal with it. I'll try to work with both. Most people are on the Facebook chat. Okay? Yes, Clementine, I can see you. I can see your comments in the poll. Okay, so I'm going to pull up the slides for today. Let's see here. This is so very awkward, but I think I'm getting a hang of it. Okay. Oh, people are still coming in. Yes, I see you. Leah McGinty, yes, so you're here. And let's see, Hans is here and Charlotte. Okay. Charlotte, the slides for chapter 12 have been posted. Okay, can you see the slides for today? It should say well, it's one of the figures that I asked you guys to study. Okay, let's see. Let's start. So let's review from here. These were the figures I asked you guys to look at. So let's start here. So we talked about different types of mutations that were created to help understand the role of the various components of the LAC operon. And we're looking here at an F prime plasmid, in a cell that creating what is known as a partial diploid or a mirror diploid. In this case, well, on the chromosomal DNA, we have a normal operator, so that's O+, LAC-I- repressor, an inactive repressor protein being produced from the gene on the chromosome, and a mutant permease, that's LAC-Y, mutant permease. And here on the plasmid end of the genetic information, we have a normal repressor, normal operator, and mutant LAC-Z, normal LAC-Y, which is the permease. And it says this overall system, the repressor plasmid, the repressor protein, which is expressed from the plasmid, can diffuse through the cytoplasm and operate on the chromosomal DNA. It operates on the chromosomal DNA. As a result, the overall system in this cell is said to be inducible because you have a repressor that is still able to bind the inducer, which is lactose, if it enters the cell. So this system, this overall cell, the system, I would say in this cell overall, it's said to be inducible. So it's inducible for beta-galactosidase on the side of the chromosome. It's also inducible for the synthesis of permease on the side of the F prime plasmid. But It's not inducible for LAC-Z because LAC-Z is mutated. So this shows you how the LAC protein acts in trans. Although there's a mutation on the bacterial chromosome for the LAC repressor gene, because the LAC repressor protein acts in trans, it can act both on the plasmid operator and on the chromosomal operator. Okay? So this system is said to be inducible. Okay. Next, we have the LAC-IS. That's the super repressor. And if you recall, the super repressor, although it's able to diffuse, it can diffuse because it acts in trans. It is unable to bind the inducer. Okay, unable to bind the inducer. So because it's unable to bind the inducer, this system is said to be constitutively off. It's turned off all the time. So there will be no LAC-Z or LAC-Y being expressed from either the chromosomal section of the genome or the plasmid section of the genome, because this super repressor will bind to the operator on the plasmid DNA, and it will also bind to the operator on the chromosomal DNA. Okay, and here's a third one. This is a LAC-OC mutation, and operators, like promoters, are cis-acting elements. So there's no diffusion here. These are not proteins. This is the DNA, so there's no diffusion here. So these are this is a cis-acting element. And in this case, it's interesting because you have LAC-Z is constitutively expressed, okay, on the plasmid end. So this is the plasmid here. We have a repressor protein that will bind to the operator site, okay, bind to the operator site, and will block. It can be inducible on this part of the genome. So on the genome part, it's LAC-Z is mutated, so it's not being produced, but LAC-Y is inducible. That's the permease is inducible. But on the chromosomal side, because you have this OC mutation, which only affects the genes that are adjacent to it, the genes that are next to it. So it only affects the LAC-Z and the LAC-Y structural genes that are adjacent to it. This OC mutation has no effect on the genes found on the plasmid, because an OC mutation is a cis-acting mutation. So it only affects the genes that it's next to. Okay, so on the side of the chromosome, LAC-Z will be constitutively expressed. There will be no LAC-Y because it's mutant. Okay, so it says LAC-OC mutation affects expression of genes on the same DNA as the mutation. Okay, so let's go to our questions. But before I do, I need to know if you're seeing this slide. The slide, it should say inducible, question mark, constitutive, question mark, or unable to express, question mark. Let me know if you're seeing that. Okay, great. So here are the questions. Okay, so Talissa, you're seeing it. Okay, so if Talissa is seeing it, I'm assuming everyone else is. Okay, Abraham too. Great. So here are the questions. I will show you a genetic configuration that represents both the information found on the plasmid and the information found on the chromosome of that cell. And I want you to tell me if the genes are inducible, if they're constitutive, or if they're unable to be expressed. Okay, so that's the question. So here's the first. we have here on the left side, this would be our F prime plasmid, and on the right side, we have our chromosomal DNA. So you're seeing here on the F prime plasmid has a wild type repressor, wild type operator, mutation in the beta galactosidase gene, wild type permease on the F prime plasmid. On the chromosome side, we have wild type repressor, an OC mutation, and wild type for Z and Y. So the question is this system that we're looking at here, this cell, the genes that is beta galactosidase and permease, are they inducible? Are they constitutive? Or are they unable to express? Let's start first with beta galactosidase. In this cell, is beta galactosidase constitutive, inducible, or unable to express? And I'll wait for your answers. Okay, so Abraham, you're saying that it's repressible, and Charlotte is saying you cannot see the slide. Okay, I will post the question on Facebook. Let me put it on Facebook, on our chat. What happened? Okay, so Charlotte, that's the question. Let's see. So the question was, is it constitutive, inducible, or unable to express for beta gal? And the correct answer is, it is constitutive for beta gal. So most of you got that. Rayford, you said unable to express. Rayford, can you tell me why you thought it was unable to express? Yes, it is constitutive for beta gal specifically. Okay, what about permease? Is it constitutive, unable to express, or inducible for permease? Not inducible for permease. Let's see. For permease. Remember that there is an OC. Okay, no one got the right answer so far. I'll wait a little bit longer, see if someone could pick it up. If someone comes up with the right answer. It is OC. You have to look at the chromosomal side. Okay, let me enter it in the chat. Yes, Rayford was still on the first. So on the first one, I don't know if you guys are seeing what I'm seeing here. The first one, it is constitutive for beta-gal and constitutive for permease. Okay, let's see. Let me try to explain this a little better. So on the, let me pull this up. Okay, so why is it constitutive for both? Let's first look at the plasmid side. On the plasmid side, it would have been inducible. It would have been inducible. However, beta gal is next to a wild type operator. It would have been inducible, but on the plasmid side. But on the chromosomal side, it is constitutive. So although it may have been inducible on one side, on the plasmid side, overall in that cell, you will always have beta gal being produced. Okay, although it could be shut down on the plasmid end, there will always be beta gal in this cell because of this constitutive mutation. You will always have beta gal present. That is why it is constitutive for beta gal and also constitutive for permease. On the plasmid side, the permease is inducible, so you could turn it on and off. But on the chromosomal side, it is constitutive. So if you should ever extract, get this cell and open up, break it open and look at the contents, you will always have permease and beta-galactosidase being produced. Okay, so it is constitutive for permease for both. Let's see, Rayford, you asked the question. You said it's constitutive for permease because the repressor can't repress or bind to the operator. Correct. The repressor is unable to bind to the operator on the chromosomal side, which will affect the overall content of that cell. Okay, let's try a second one. Same question, starting with beta gal. Is it constitutive, inducible, or unable to express beta gal? Again, you have to look at the cell as a whole. Okay. Yes, you look at the individual components, but what would be the overall content of that cell? What would be the overall phenotype, I should say, of that cell? So again, is it inducible, constitutive, or unable to express for beta-galactosidase? Okay, constitutive for beta-gal is correct. Okay, so yes, it is constitutive for beta-gal and inducible for permease. Okay, if no questions, here's the third one. So here's the third. Take a look at that. Same question. Inducible, constitutive, or unable to express. Starting with beta-gal. Yes, inducible for both. Okay, let's try two more. Let's try two more. Okay. All right. Same question for number 4. P is a promoter mutation. So P is P minus is a promoter mutation that prevents RNA polymerase from binding. Okay, it is constitutive for permease. No expression. There you go. Okay, correct, Rayford. So yes, constitutive for permease, no expression for beta-gal. Let's see why. So beta gal is under the control of this promoter, and it's a mutant promoter. And so if RNA polymerase is unable to bind the promoter, you will not have expression of any wild type form of beta gal. But on the DNA, on the chromosomal side, you have an OC mutation that would allow constitutive expression of the permease, which is wild type on that side. Okay, last one. Okay, yeah, no expression for beta gal or permease, unable. Okay, let's move on. Okay, let's move on. So we spent quite some time here, but I thought this was important for you guys to do. Okay, we'll move. All right, so let's move on. Let's look at, we're running out of time, so I'll skip some things and look at how, another way this lac operon could be regulated. So the lac operon is regulated by lactose, right? So if lactose is present, then you will have expression of these structural genes. But it can also be regulated by glucose. Can also be regulated by glucose. Let's see. Are you guys able to see the slide? My current slide? Okay, so I was saying that the lac operon is regulated by lactose, but it's also regulated by glucose. So when both glucose and lactose are present, the cell will preferably use glucose instead of lactose. So if both are present, the cell will choose to use lactose versus using, would choose to use glucose rather than use lactose. And in that case, when glucose is present, because of that, the reason being, when glucose is present, the genes for the lac operon are turned down. So how does this occur? Well, it occurs in the following way. When glucose is present, let's start with, let me see here. Yeah. So just to be, let's start off with when lactose. When lactose is present, we know that when lactose is present, lactose binds to the repressor protein, right? And allows RNA polymerase to bind and allows expression of the structural genes. That's when lactose is present. When glucose is present, however, this is what happens. When glucose is present, glucose causes an increase in what it is in cyclic AMP. Cyclic AMP then binds a positive regulator, binds a monomer known as cyclic AMP receptor protein. So it binds these receptor proteins, forming a complex, a positive regulation complex. This complex then binds to the promoter site and prevents the RNA polymerase from binding to the promoter site, and hence it will stop production of the structural genes. So in the presence of glucose, the lac operon is repressed. So glucose acts as a repressor of the lac operon. Unlike lactose, lactose acts as an inducer of the lac operon. So glucose is said to be, this type of repression is known as catabolite repression. And let's see, let me go over this slide here. It says here, this complex, the CRP cyclic AMP complex makes direct contact with the RNA polymerase. So it makes direct contact with the RNA polymerase. And without interaction of this complex, the RNA polymerase can bind to the promoter, but is less likely to unwind the DNA and initiate transcription. So this complex, this CRP complex, binds to the polymerase and it enhances transcription. Okay. I think I'm confusing you guys. I think, let's see here. Yeah, so the CRP cyclic AMP complex is a positive regulator of lac transcription. So it's a positive regulator of lac transcription. But when glucose is present, this complex does not bind and enhance transcription. So as a result, the transcription of the LAG C, Y, and A genes are turned down. And I think this is what it's showing you here, okay? So glucose, it says here, glucose decreases adenyl cyclase, okay? So when glucose is present, you don't have this complex being formed. And if this complex is not formed, then you don't have enhancement of transcription from the structural genes, okay? And as a result, this will be turned down when glucose is present. So this complex, the CRP cyclic AMP, acts as a positive regulator it enhances transcription of the LAC C, Y, and A genes. Okay. Let me know if I confused you. Should I go over this? I think I did confuse you. Should I explain further? Or attempt to explain again? Let me know. Okay, confused. Great, let's try this again. Okay, so this complex, the CRP cyclic AMP complex, it says it makes direct contact with RNA polymerase. Okay, so it binds to RNA polymerase, and that complex of the cyclic AMP, the CRP and cyclic AMP binding to the RNA polymerase enhances transcription of the structural genes. Okay, it says without interaction of that, then the polymerase, it will bind to the promoter, but it doesn't initiate transcription very well. Okay, so the CRP cyclic AMP complex acts as a positive regulator of transcription from these structural genes. This complex, that's the cyclic AMP CRP complex, will form only if glucose is absent. Okay, in the absence of glucose, the complex forms and enhances transcription of the structural genes. When glucose is present, this complex does not form, and so you will have no transcription of these structural genes. So glucose, in essence, as a result, glucose decreases expression of the structural genes for the lac operon, and the cell will then use glucose instead of lactose if it's present in the media. So bacteria have a preference for using glucose. If both glucose and lactose are present in the media, the bacteria will choose to use glucose instead of lactose, because glucose, when present, shuts down the production of this positive regulator complex. That's the CRP cyclic AMP complex. And if there's no production of this cyclic AMP CRP complex, then you don't have enhancement of transcription from the LAC-Z, Y, and A genes. Okay, let me know if this is clearer than previous. Okay. Great. All right, so let's move on. Well, we don't have time. We actually have 2 minutes left. And what I will do on Friday is look at another operon, which is the tryptophan operon, and which is an example of an anabolic operon. So lactose is one that produces genes that allows for catabolic breakdown, or breakdown of components or molecules. Anabolic synthesis refers to the buildup of molecules. That's the trip operon. So we will start here on Friday, okay? So let me get back. Yeah. All right, so I will see you on Friday, or you will see me on Friday. I will look at, we'll start by looking at the regulation of the tryptophan operon, which is an example of an anabolic operon. Okay? So I will see you.